I'm one of your hosts, Jonathan Ullman, alongside the Mickelson Prize laureate, Tom Kea.
What's up, John?
How are you?
Good. How are you?
Good. I wanted to call you a laureate, Tom, because that's
what they're called.
That's
right?
what they're called. It's a fun term, laureate, laureate. I feel like
what does it really mean?
I have no idea, to be honest with you.
Should we just Google it?
We can. I have a feeling that it's just like a recipient of some sort, but I feel like Rick from The Walking Dead. I say laureate, Carl. You know,
a person who is honored with an award.
Yeah. So a recipient of an award
for intellectual achievement. I should get one of those.
Yeah, you got one from Mush.
Oh, thanks.
The Mickelson Prize laureate, Tom Kea. How are you?
Great, man. Episode 35. We've come a long way, man.
We've done it. This is it. This is the last episode of trèSci Weekly. We said once we get to 35,
that's it.
We're calling it, throwing in the no, I'm just kidding. Thank you guys for joining us today. Today is a very special episode. It's the Columbus Day episode.
As they said, back in 1492, Columbus sailed the ocean blue so we could have a day off school.
Exactly. That's nice.
That's from South Park.
Is it?
Yeah.
Nice. No, nothing really too exciting about this episode, other than the fact that we're going to talk about the 2017 Nobel Prize laureates, Tom.
Yes.
It's going to be the focus of today's episode. So, pretty groundbreaking research that we're going to be talking about. Pretty good stuff.
Yeah, definitely. Exciting things.
If you're just joining us, thank you so much for tuning in. We are happy to have you here with us. It is trèSci Weekly. It's our weekly video series on science news, science trending topics, and different research discoveries.
Yep. We can talk about anything science related.
Literally anything. Anything at all. And then some things that aren't science related, I'm sure, will slip in there. You can, of course, watch this show live with us at 8:30 p.m. on Monday nights. Chat with us in the chat, and we're happy to have you here during the live recording of this. If you can't catch us for the live show, ain't no thing, ain't no problem. You can always watch the free replay on our YouTube channel. And if you like what you see, we would be honored for you to subscribe to the channel. That way you get the little alerts for when we go live, and
grow the community.
Exactly.
Love science with us.
Part of the trèSci community here. You can also go to facebook.com/trèSci Weekly and like us on that platform. You can go to twitter.com/trèSci Weekly and follow us on that platform. And if you're feeling extra special generous, you can go to patreon.com/trèSci Weekly and throw us a few bucks to help support what we do here on the stream.
Sounds good.
Tom.
So as you mentioned, we're talking Nobel Prizes.
We are talking Nobel Prizes. Since this is a very Nobel Prize research-oriented episode, was there anything outside of that you saw during the week that you want to give a little honorable mention? Sometimes we like to do that.
Yeah, we were following Hurricane Nate, which made landfall. I think it made landfall as Category 1, almost Category 2.
Yeah.
There was some storm surge, coastal flooding, and then it sort of combined with another low-pressure system. If you look at it on the radar, it sort of morphs into another one, and it's a long band of rain that's over us right now.
Mm-hmm.
So it kind of merged. So I was following that story a little bit.
Correct me if I'm wrong, Hurricane Nate broke the record for the fastest northward motion in the Gulf, right?
I think it might have.
Yeah, it was moving quick.
If you think about it, when we followed Maria and Irma, right?
Yeah. Those
were really slow moving. This one swept right, made landfall quick, and now it's all the way up here.
Yeah.
Way different than those other ones.
Yeah, cruising.
Yeah. Yeah, a little bit smaller overall. It wasn't as formed when you looked at it in the radar. Sometimes it looked like it was only half a hurricane. But yeah.
Yeah, still some considerable damage.
There's a lot of tornadoes too.
Really?
Which is a big deal.
I saw some videos of water spouts.
Yep. Yeah, I've seen those too,
Yeah, those were pretty exciting, I guess.
Yeah, terrifying and exciting all at the same time.
Yeah.
Anything else?
I want to give a quick shout out. We were just talking about this very briefly. The first American Space Council meeting took place over the course of the week. Vice President Mike Pence very authoritatively said that we're going to put American astronauts back on the moon.
Yes, he did, yeah.
And that we're going to go beyond the moon to Mars. And that's like a huge initiative and priority for the Trump administration. And I think that's
pretty cool.
That's pretty cool. I want to just throw out there really quick, Tom. I saw this on Twitter. I watched the video. I saw his tweet about it, and I thought to myself, "Wow, cool! I'm gonna, like that tweet, retweet it, right? Mm-hmm. That's what you do on the Twitters."
Yeah.
And then I read all the comments below, and it was just like, everybody just hating on Mike Pence, just bashing him. And I'm like, all right, I get it. He's Mike Pence. He doesn't do everything right. He doesn't do a lot of things right, and you don't have to agree with him on everything, but what did he say here that was so offensive that you gotta hate the man? I feel like people gotta be better at picking and choosing the things that
offend them and
right,
you need to call out.
Yeah, and you don't have to hate everything that comes out of a person's mouth just because you don't agree on some things with them, right?
That's a good point to make.
So we're not going to discuss too much about that on today's show, even though that did happen in the past week, along with Hurricane Nate. Because we really want to focus on the
Nobel Prize.
The Nobel Prize, and specifically the Nobel Prizes in the sciences, in the hard sciences, right? Because there's also the Peace Prize and the Economics Prize.
The economics today too, yeah.
Yeah, and the Literature Prize. Those are all wonderful.
We're trèSci Weekly. We're not Trey Literary.
Trey Lit, yeah. Trey Lit. Kyle's Trey Lit, but that's about it over there.
All right.
So let's get on into it. I figured we'd just go in order as they were announced, right? So. Yep. The 2017 Nobel Prize laureates have been announced. The recipients have been chosen by the Royal Swedish Academy of Sciences for the Nobel Prize in Physics and in Chemistry, and by the Nobel Assembly at Karolinska Institute for the Nobel Prize in Physiology or Medicine, which will be the three we focus on this show. The announcements rolled out day by day starting on Monday, October 2nd, which was the Nobel Prize in Physiology or Medicine, which was awarded to Jeffrey C. Hall, Michael Rosbash, and Michael W. Young, quote, for their discoveries of molecular mechanisms controlling the circadian rhythm, end quote. And we have a little graphic to put up of the three Nobel laureates there. This is comes by way of nobelprize.org, and the illustrator here is Niklas Elmehed. So we'll get the other ones are also illustrated by him as they go. Just want to give credit where credit's due there. Tom, before we begin discussing this really in depth here, what is circadian rhythm? What are we talking about when we're talking about circadian rhythm?
Yeah, so I actually was reading the article said it what circadian stands for, the Latin roots. I can't remember right now off the top of my head.
Wait,
something around daily, circa.
Circa is around, and
Adian is
dia day, around a day. Is that
is
that right? Something along those lines.
Basically saying that there's a pattern to the cycles within our own bodies based on the amount and timing of the sunlight that we receive here on Earth.
Mm-hmm.
So I don't know if you're aware, but it gets dark at night, and then during the day it's light out.
You're changing my world here.
And you kind of expect that there's some sort of relationship that human beings and animals have with the cycle of the moon and the sun, considering all of our energy comes from the sun, and that there's that intricate link between the human beings, organisms, and their environment.
Right.
So what we're really talking about with this one is that it just helps regulate the chemicals in our body, our hormones and things, based around the cycle of the day. So depending whether you're awake when it's light out or if you're sleeping at night, that's the ideal way, right?
Right.
But depending on whether you sort of fall off that a little bit, or you are awake late into the night, different things can affect not only like how you're feeling, like you didn't sleep enough, but literally affect almost like the underlying premise of your body chemistry, where that it's been evolved here on Earth, right? So it makes sense that you have to go through this cycle.
Yeah, and I guess the thing with it, right, is that it's built in. It's not just so much like, well, you know, we should be awake during the daylight hours and we probably should sleep at night
and
because that makes
all
sense, right? There's actually mechanisms within our bodies that are regulated on this cycle, right? And
yeah, that's a good way to put it.
And these gentlemen discovered those mechanisms at kind of a cellular level, right?
Yeah, just describing exactly what's going on with the hormones, the proteins that they make, and what the effects of those proteins are.
Cool.
That's what they've talked about.
All right. So let's jump on in. Let's get into the nitty-gritty here a little bit. So I have written a little synopsis based on what I researched and what I've been reading about it. But we'll also jump in. I'll be quoting some of the press release that the Nobel Prize website put out for, more specifically, what the contributions of each of the three gentlemen that were awarded the prize, how they fit into, the grander scheme of this research.
Okay.
All right. So let's jump in. While the idea of an internal biological clock has been around for at least a few hundred years, the mechanism by which it works has only recently been discovered. The groundwork was laid back in the 1700s when Jean-Jacques Dortus de Mairan, I think I'm pronouncing that correctly, noticed the leaves of mimosa plants would open toward the sun during the day, but close at night. His experiment showcasing that the mimosa plant continued to do this even when in constant dark environments suggested that organisms have some sort of endogenous or internal clock.
So this is dating back all the way to the 1700s.
Mm-hmm.
So pretty cool how you can follow the science throughout the centuries.
Yeah.
As well as that just the experiment itself, how the plant still opens up regardless of it being light or dark. It just has the timing down. It knows sort of how fast our Earth, well, it doesn't know how fast our Earth revolves, but I'm sort of just putting it that way.
Right.
It's aware of the cycle of day and night, independent of whether it's actually day or night.
Right. So again, yeah, so we're looking at this from a standpoint of like, well, it's not the actual light that causes the leaves to open up, right? The plant just knows the cycle.
It knows when the light will be there.
Right.
So it has to open up to get it.
Right.
It doesn't matter if it's not sunny that day, it knows the time period of the cycle.
Yeah.
Pretty cool, yeah.
Pretty cool. I mean,
it's a plant.
Probably a breakthrough experiment in the 1700s.
Yeah.
Way back when. Flash forward to 1971, where Seymour Benzer and Ronald Konopka conducted an experiment to try and trace a genetic origin for the behaviors associated with circadian rhythms in fruit flies.
Fun fact, fruit flies are one of the most experimented on organisms.
Mm-hmm. Mm-hmm. I have to mention this. I saw a tweet that I guess back in the day, man, what is her name? Alaskan woman who was the candidate for
Sarah Palin?
Sarah Palin, thank you.
Yes, I got it.
Sarah Palin made a comment back in the day that she was like, yeah, we do all this extraneous spending on science research, and people are literally just researching fruit flies. Can you believe that? And somebody took that and made a meme out of it, and it was just like fun to basic science, because now this is a Nobel Prize being awarded for research done on fruit flies, which I think is funny.
Yeah, they're just an extremely simple organism that you can use as a model to extrapolate science. It doesn't matter that it's a fly. It's like the model for your experiment.
Yeah.
But I see where you can get that misnomer about it.
Totally. Benzer and Konopka looked at mutant fruit flies, ones that would carry out pupal eclosion, which means emerging from the pupa. Right? pupil occlusion, and locomotion activity, or just moving around in layman's terms, at arrhythmic, shorter than daily cyclic periods, being 19 hours, and longer than average cyclic periods at 28 hours. So they were like, all right, well, we can look at what fruit flies do in terms of their daily routine, and then we'll find some fruit flies that do this kind of off, that like arrhythmic, there's like no rhythm to it really whatsoever, and then they have some that operate on a 19-hour cycle instead of the daily cycle, and then they have some that operate on a 28-hour cycle versus a daily cycle. And they said, all right, let's hone in on these ones that do it kind of oddly, and let's see if we can trace genetics. Let's see what we can figure out with this.
Yeah, so it seems like a fruit fly is actually like an ideal organism for this because it has these different cycles of circadian rhythm, which I don't think every organism has.
No, probably not. I mean, mush sleeps all day, constantly, so there you have it. With genetic markers, all 3 different types of mutant fruit flies showed mutations in the same gene, which was later named period. Probably not the best choice of name for a gene, because this is going to be a little confusing as we continue on now, as I'm using the term period, but not the definition of period. Yeah. But whatever. We'll forgive them for the naming convention, I guess. Enter our 3 Nobel laureates, who in the mid-1980s were able to isolate and sequence the period gene, providing the first look at its molecular structure. This was all fine and good, but the mechanism for how the gene operates on a daily cycle was still unknown. The first clue came by looking at PER, the protein produced by the period gene.
So the period gene makes a protein that they refer to as PER.
Correct.
Okay.
PER would accumulate during the nighttime in the fruit flies and degrade during the day. This fact would ultimately be characterized by the transcription-translation feedback loop. And this, Tom, I believe, is the breakthrough that the Nobel Prize probably hinges on, right? Is how exactly this feedback loop works. We have an image here that we can go over to try to explain it a little bit. So the transcription-translation feedback loop starts in the cells, right? We have number 1 up top here. This would be kind of your everyday cell that has some cell membrane, some kind of cytoplasm,
where all the stuff's floating around,
nucleic acid in the middle, and then a nucleus in the middle, and there is a DNA strand here that a messenger RNA will begin the transcription of, and that gets triggered by sunlight, right?
Yeah, so that only happens during the day. Yeah.
Correct. So the morning time, maybe, I don't know, afternoon, this messenger RNA is prompted to go copy the information on the DNA strand in order to produce a protein, right?
Yep.
And that's what we see here in number 2, is the messenger RNA produces now the PER protein from the period gene.
Yep.
Right? And then, Tom, it continues to do this. It builds a whole bunch of PER proteins. So much so that they start to accumulate, and by stage number 4 here, where now we're entering the nighttime, there's so many PER proteins that they build up and effectively shut off production of more PER proteins. Mm-hmm. So it creates so many that it has to stop creating them. And that, I think, is kind of the basis of this feedback loop here, because those PER proteins will then, over the nighttime, begin to degrade and go away. And then by the time we reach morning again, it's time to trigger the whole cycle all over again, right? And so that's how this ultimately fits in with a 24-hour daily circadian rhythm, right?
Yeah.
This is how our cells operate on a daily pattern, not just in response to sunlight, but
response to the period of sunlight that you typically receive.
Exactly.
So I think, just to clear it up, I think you might have said it backwards when you mentioned the definition that they accumulate during the night and degrade during the day. The PER.
Thank you. You're absolutely right. I did mess that up. Right. So
they accumulate while you're sleeping or away from the sun, and then they degrade while you're in the sun or during that period.
Right.
So it doesn't actually matter if you're in the sun, it just matters during that time period.
Exactly.
I keep saying it, like, because during the day it's sunny, typically.
Yeah.
You know, but it's just that time period that the biology is built around.
Right. And that's the fascinating part of it, is that you don't even really need to be in sunlight. It's not a response to the light itself, I don't believe. It's a response to the fact that there is daylight.
Cycle of day and night.
Right. Now, of course, Tom, this is just a simplistic model of that feedback loop. And in practice, when these Nobel laureates, came up with this sort of idea, tested it out, and then looked further into it, it's much more complicated than that.
Okay,
right?
yeah.
There's like the timeless gene and time proteins that hitch a ride on the PER proteins, and
yeah, I think there's like 3 of them or 4 of them.
Yeah.
That go up and down depending on how much of one or what time you're in, and it's a complicated cycle.
Yeah. And basically, I think it means that the cycle has checks and balances, which would make sense, right? You don't want your circadian rhythm to just get thrown off by,
if you just stay up all night for
Right.
a week or two.
Right.
Your body, it can adapt and it can balance out.
But that being said, how does this relate back to medicine and health and physiology here? So, according to the advanced press release by Carlos Ibanez, not the baseball player, that was Raul Ibanez. Carlos Ibanez was a professor of neuroscience at Karolinska Institute and member of the Nobel Assembly. There was a paragraph here of kind of like, what are the impacts of this research? So, quote, "Chronobiology has an impact on many aspects of our physiology. For example, circadian clocks help to regulate sleep patterns, feeding behavior, hormone release, blood pressure, and body temperature." And we have an image of that as well, kind of a little graphic that shows how much of your body depends on these circadian rhythms. And credit goes to, of course, NobelPrize.org and Mattias Karlen for this image. Molecular clocks also play critical roles locally in many tissues. Ablation of clock genes in animal models result in arrhythmic production of hormones such as corticosterone and insulin, and this was a study done by Sun et al. In 2008. Clock genes also exert a profound influence on metabolism through the control of gluconeogenesis, insulin sensitivity, and systemic oscillation of blood glucose. And this was a study conducted by Panda in 2016. Evil pandas conducted this study. Can you believe it? Sleep is vital for normal brain function, and circadian dysfunction has been linked to sleep disorders, as well as depression, bipolar disorder, cognitive function, memory formation, and some neurological diseases, as researched by Gerstner and Yin in 2010.
So I just wanted to talk a little bit about that.
Yeah.
It's cool that we're seeing how there's multiple studies by many different people and groups of people that have given us more knowledge about the circadian rhythm. And then the other point I wanted to bring up was that they're saying that along with the circadian rhythm and sleep, it's vital for your brain function, and as well as it can affect you when you have depression or bipolar disorder. And my question to you would be, do you think that disruption in your circadian rhythm can cause things like depression and bipolar disorder? Or once you start getting depressed or have bipolar disorder, does that have an effect on your ability to like sleep and have normal circadian rhythm? You see what I'm saying? Does
Yeah.
I can see it almost kind of going both ways, kind of a little bit. It seems like they're connected in some way.
I would agree.
Like as you get depressed, a lot of people have a harder time sleeping once you're depressed.
Absolutely.
And then that messes up your cycle, which could then make your depression worse by just making you a less healthy person overall.
I wouldn't be surprised if the answer is one. If your circadian rhythm, if the cycle is disrupted, and that leads to kind of behavioral trends that would lead towards depression, I think then that depression probably makes it more difficult for you to get the rhythm back, right? To get your balance back.
Yeah, so then you could sort of have a chemical imbalance in causing you depression from having your circadian rhythm be out of whack.
Yeah, and I think that's why you can look at things like depression and sort of see how they can spiral out of control, is because it's probably very difficult to come back from being off of your normal cellular rhythm and then experiencing kind of this new rhythm of how your body reacts to
new conditions.
Right, new conditions of alcohol content, and just how your body reacts to that in general. Your blood alcohol level increasing at this time of day and then maybe decreasing at this time of day. It's probably difficult then for it to just jump back into a normal routine as it was. Yeah, it's a great point though. It's kind of a chicken and the egg question. Like what comes first and what leads to the next thing.
But you can sort of see it happening, right? Say you're a student and you just go to college and now you're staying up all night, like, because now you got to study more and do more homework and meet more friends and things, and then out of nowhere I could see how just that one thing of changing your rhythm by even a few hours could throw things all out of whack, kind of.
Yeah. I mean, one of the more kind of classic examples of your circadian rhythm being thrown out of whack is jet lag, right?
Yeah, that's a great example.
When you travel out of...
you got a few hour gap.
Yeah.
And that doesn't make sense.
Right, you travel out of your time zone and now all of a sudden, it's jump ahead 6 hours. And
it's more than just losing 6 hours of sleep.
Right.
It's because you're 6 hours out of phase of your normal rhythm.
Right.
Yeah.
And
that takes time to adapt to
I can speak from personal experience. Like it makes you feel bad, right?
Yeah.
Like it's not just like, oh, I'm tired. You feel like, wow, I'm so out of whack right now. Like everything is just mixed up, and it takes a few days to kind of get over it and adjust and get to a new rhythm, right? What'll be interesting, I think, is to see, do we jump in to a pharmaceutical solution to this?
And try and regulate it?
Yeah. Is there a way to take a drug or some sort of medicine to help with something like this?
That keeps it as regular as possible. Like fiber is for your metabolism.
Yeah.
You take this supplement for your circadian rhythm.
Yeah.
Who knows?
I don't know. And the other thing that I thought about this was, we talk a lot about, especially on the last episode, traveling to other bodies in the solar system.
We were just talking about going to the moon and Mars.
Right. And so...
Yeah, I could only imagine. If you get jet lag going to Hong Kong, what kind of profound effect would you have going to Mars, right?
And this is an evolutionary thing, right? So this is our cells and our bodies adapting to the day, being a 24-hour day. Now, if we take people and move them to Mars, where the Martian day is no longer 24 hours... I think it's something like 28 hours long.
Like about a 4-hour difference.
Will we always be out of whack with our circadian rhythm, or would we be able to adapt to a new length of day?
Yeah, that's a good question.
And how much would that affect us?
Yeah, I think it's tough to say, right? Because it's chemical and it's biological, I think there's a good amount of like plasticity to the whole system in general. Like it can sort of flex and change a little bit. Like we see a lot of human beings like work overnight shifts for like 20 years. And so you can see that it can be stretched and bent, but I don't really know how it would respond to being on a completely different planet like that. I don't know.
Yeah. Yeah, it's
tough to say, yeah.
I mean, we won't know until we try it, and we may be trying it in the next decade. Who knows?
Yeah, right.
So that'll be interesting to see. Laura Haller in the chat, one of my cousins down that that I just went out to dinner with.
That's cool.
Said currently fighting with jet lag right now, and that, like I said, jet lag is not just being sleepy from
for a few hours.
Yeah, it's a little bit more intense than that. So I hear you. Our bodies are fickle beings. They can definitely get thrown out of whack pretty easily there.
Thought just pops into my head.
Go ahead.
What if you're in a binary star system and you have like light for 6 hours, darkness for 6 hours, light, do you think it's a necessary part of evolutionary biology to have some sort of rhythm, or is it sort of just like a happy medium that our bodies were like, okay, it's light here, it's dark here, we kind of get this cycle, and then it sort of became in, or is it like inherent to being able to live in that environment? I mean, I don't think you could definitely answer that, but that's a hard question to have a definite answer for.
We do know that there are planets that are confirmed to be able to orbit binary star systems. I believe that is something that we've confirmed. If there were, let's say, life on this planet where there's a binary star system, you would expect that evolution would drive a circadian rhythm to respond to
that environment,
that light being received. And if so, then as long as there's nothing completely irregular about that system,
if there's some sort of regularity to the period,
right, then it would be probably fine, I would think, unless, again, that life form moves from one planetary body to another that has a different rotation period, and then it gets thrown out of whack. But that's assuming that evolution works consistently across all life forms, which we have no idea.
As well as it being consistent in its own nature, because it's kind of inherently random to its core at some point.
Right.
So yeah, a lot of interesting questions that it brings up, as well as it gives us a lot of insight into our bodies, as well as just raising even more questions, which I think that's a sign of good science, right?
Yeah. Carrie Kalita in the chat says, people running the Mars rovers like Joel stay in an environment that goes on a sol, or a Martian day. And then he goes on to say, otherwise they would be off time on an accumulating error, day would be night, right? So they would constantly be out of sync with the data that they're trying to collect from the Martian.
So they just pretend as if they're on the Mars surface.
Which Tim Glotch actually mentioned to me too when we were, or I guess we took it at separate times, but when I took the Geology of Mars course with him, he mentioned that was something that he had to go through. That's fascinating, to be on the schedule of another planet's day and night cycle, but living on Earth.
Yeah.
I can only imagine how difficult that must be, and I would love to talk to either of those guys, Joel or Tim, about that experience in the context of circadian rhythm, and pick their brain about it. Shall we move on?
Sounds good.
All right. On Tuesday, the Nobel Prize in Physics was divided. One half awarded to Rainer Weiss, the other half jointly to Barry C. Barish and Kip S. Thorne, quote, for the decisive contributions to the LIGO detector and the observation of gravitational waves, end quote. And of course we have the illustration from nobelprize.org and Niklas Elmehed again for that one. So let me bring that up for us as well. These three gentlemen.
Nice.
And now, Tom, we've talked quite a bit about gravitational waves on trèSci Weekly.
I'd say like 4 episodes maybe.
At least, and we
just because they're cool, as well as we had Tyler on the show.
That's right.
Who sort of gave us a little bit even more insight to them.
Yep.
As well as it just being sort of a new field where they're having new discoveries that are relevant.
Yeah, and in fact we just brought gravitational waves up last week, on last week's episode.
Because they added the 3rd detector, right? They added the...
Well, that was a little while ago, and then last week we talked about the 4th...
Oh, yeah, the 4th
detection.
Yeah.
Right? So they've been hot in the news, so to me, not a huge surprise that the Nobel Prize was awarded to them. But
I didn't really think about it in my head, like try to make a prediction, but it makes sense, after seeing it.
Yeah, and I think it's a nice opportunity for us to, again, kind of reflect on how important this is. I know last week I said with the 4th detection that I was surprised at how much attention was given to it, I think, because, first detection, wow, amazing. Second detection, wow, cool, great. Third detection, awesome. Fourth detection, all right. Like, do we need to be making this big of a splash about it every time it happens? But the answer kind of is yes, right?
Yeah. It might lead to some really interesting science relatively soon. I just watched a video where they interviewed Kip Thorne, and he was saying that it's more than just the fact that they detected 2 black holes coming together and sending ripples across space-time. What he said, what he's most hopeful for, is that he wants the research to be able to bring together quantum mechanics as well as gravity in a uniform theory, and he's hoping that understanding the fabric of space-time more would give more data and knowledge to the people doing string theory and M theory, who he thinks, as we get more data from gravitational waves, will be crucial in them creating like the super unified theory of physics currently, that gravity doesn't really make sense in terms of the other forces.
Right.
So he's saying that hopefully down the road that this is what this sort of research leads to, not just the fact that we want to detect these things.
Right.
Yeah, and there's things we can learn from them.
Yeah. And then factor in but we do want to detect what we detect. It's like,
we want to prove that we can at least find them.
Yeah, and from an astrophysics perspective, it's like really interesting that, these exotic things that are happening in our universe can actually be studied in some tangible way now.
Yeah, over 1 billion light years away. Amazing. Losing the mass of multiple suns in an extremely short amount of time and having so much energy to ripple across the cosmos and reach us.
Yeah.
That's sort of a profound idea in itself, even if not much comes from it.
Yeah, it's incredible. So let's jump in a little bit more in depth here. Following the announcement of the 4th confirmed detection of gravitational waves resultant from the merger of black holes located some billions of light years away, the contributors to the development of LIGO are certainly well deserving of their Nobel Prize recognition. The detectors at LIGO have simultaneously vindicated a 100-year-old prediction made by Albert Einstein within the general theory of relativity, as well as providing a brand new, unique way for astrophysicists to probe the far reaches of the universe for extreme phenomena without requiring the detection of light.
And I think that's an important point to make, is that in a lot of astronomical data collecting, it's almost all capturing light, right?
Yeah.
Leaving long exposures, collecting light from many, many light years away, and then being able to see them.
Right.
But being able to detect parts of the universe without even needing light, that's kind of a cool way. It's like expanding our senses to the universe.
Yeah.
Cool.
It makes sense that most telescopes and detectors that we have make use of the detection of light because, it's one of our premier senses. We detect light. We have eyeballs that detect light, and light carries a lot of information about where it came from and, what
things make it up and everything.
Right.
Based on spectroscopy of the light and things like that.
Yeah, and so now we're using, waves of gravity, waves of space-time in order to learn something about the far reaches of the universe, which is something that our senses here on Earth are not privy to whatsoever.
In the least bit,
yeah, you don't feel a gravitational wave pass through you.
Yeah, we're going to talk a little more about how precise we need to be to collect them, right?
Yeah.
So I don't want to jump into that just yet.
Yeah, don't want to spill the beans. What I do want to bring up are two images. The first one being of LIGO Hanford, right? So the Washington State detector. And I want to kind of talk about how these detectors work. I know we have talked about this in the past.
Mm-hmm.
Tyler Cohen came on the show and gave us a really brilliant description of this in the past. But I think that we'll take a look at it, and then we'll show a video as well that should help refresh everybody's memory of this a little bit. So here's LIGO Hanford, and you can see there are these two arms, right? These two long, skinny tubes, basically, that reach out from the center of the facility. And then I want to bring up the next image is an image of what's called a Mickelson Interferometer. And this is basically the physics behind what's going on with these detectors, right? This is something that I've used in physics lab at college.
Yeah.
And it's a pretty simple idea where you take a laser beam, you split it, and then you reflect it back in a mirror so that the phase of the light, right, the wave crest and the wave trough of the light matches up perfectly so that you can either get no signal back or you can amplify signal into a fringe pattern.
Okay.
Right? And so if just comparing just the way that this apparatus, the Mickelson Interferometer, looks like from a design perspective, and you look at that aerial view of LIGO Hanford, you can basically see that LIGO is a giant Mickelson Interferometer. Mm-hmm. And that's what we're using here to discover the gravitational waves. So to better explain how an interferometer works to detect these gravitational waves, we're going to watch a short video that stars actually the Nobel laureate Rainer Weiss, which is featured on LIGO's webpage. And this video credit goes to the National Science Foundation for this one. So
sounds good.
Let's throw the headphones on.
Thank you.
No problemo. And Kyle, you let me know when to hit play there, bud.
To measure the stretching and squeezing, Ray turned to a device called an interferometer. A laser beam is split and sent down a pair of long perpendicular tubes, each precisely the same length. The two beams bounce off mirrors and recombine back at the base. The light waves come back lined up in such a way that they cancel each other out.
And you add them together, you get nothing. You get a zero, a big fat zero. No light gets detected at the photodetector.
But when a gravity wave comes along, it distorts space and changes the distance between the mirrors. One arm becomes a little longer, the other a little shorter. An instant later, they switch. This back and forth stretching and squeezing happens over and over until the wave has passed. As the distances change, so does the alignment between the peaks and valleys of the two returning light waves, and the light waves no longer cancel each other out when added together in the recombined beam. Now some light does reach the detector, with an intensity that varies as the distance between the mirrors varies. Measure that intensity, and you're measuring gravity waves.
The light takes longer time in here than it did in this arm. Now it takes a shorter time, and these things don't cancel so beautifully anymore.
Love how into it he gets. It's so cool.
Yeah, I guess if you're smart enough to do the math of gravity waves, you get pretty hyped on it.
Yeah, absolutely.
So a lot of times in the video they mention that they need to be exactly the same length and that they come together at exactly the same time. So when they say exactly the same length,
they mean exactly.
They mean like
yeah,
exactly the same length, right?
Yeah. So that's what we're going to shed some light on in just a second here. So I'd like to read another excerpt from the LIGO press document. This is given by the Royal Swedish Academy of Sciences.
Sounds good.
And this was on a brief history of LIGO and sort of a description of how the Nobel laureates fit in here. Quote, "The basic concept of a Mickelson-like interferometric detector for gravitational waves was first sketched in an article by the Soviet physicists M.E. Gerstenstein and V.I. Postuvit in the early 1960s. The idea was resurrected a few years later by Weber and his former graduate student Robert L. Forward, who constructed the first prototype gravitational wave laser interferometer at Hughes Aircraft Research Laboratories in Malibu, California, where he and his colleagues had started research and development of gravitational wave detectors."
So this is kind of cool. Another example of how the science started decades ago again.
Yeah, and you'll see, I mean, this is pretty much a trend in the Nobel Prizes that it's
not just one idea that someone came up with and then, ooh, I figured it out.
Yeah.
You know, it's years of hard work and building on theories.
Yep, and good science, doesn't start from nothing. It starts from
What we know, yeah.
Right, from very researched areas of the world. In parallel, Rainer Weiss at the Massachusetts Institute of Technology, MIT, inspired by Pirani, developed similar ideas which, in 1967, enabled him to demonstrate a laser interferometer with sensitivity limited only by photon shot noise. Decades of ambitious efforts building on Weiss's designs resulted in the realization of the Advanced Laser Interferometric Gravitational Wave Observatory, or ALIGO for short. Weiss's report from 1972 constitutes an early exploratory study of an interferometric gravitational wave antenna targeting detection of pulsar signals, notably from the crab, not the harbor crab, but the crab nebula. The Crab Nebula, and describes and evaluates the fundamental sources of background noise that critically limit the antenna performance. These include seismic noise, gravitational field gradients, noise due to thermal gradients in the vacuum chamber, thermal noise associated with the test masses or mirrors and their suspensions, amplitude variations in the laser output power, laser frequency instabilities, pressure recoil effects on the mirrors, and shot noise in the photocurrent at the output. Potential effects from geomagnetic storms and cosmic ray showers are also considered. At the end of the document, Weiss points out that a meaningful pulsar search requires a kilometer-size interferometer. End quote. So I wanted to throw that in there mostly for the list of things that affect your interferometer, right?
So we showed that video and it seems pretty simple, but they're looking for a change with like the fraction of an atom.
Mm-hmm.
So all the things that you listed, they refer to them as sources of background noise. So it's just anything that could disturb the laser. And when you're talking about a fraction of an atom, it's basically everything.
It's everything.
You have to account for every little thing that could happen.
Yeah.
And then to make sure you're only detecting the gravity waves.
Yeah.
That's why you need to take into account literally everything.
So basically, what you're telling me, Tom, is that there is one of these LIGO detectors located in Louisiana, and Hurricane Nate just blew past it, right? Just blew past... Jesus, what's the city in Louisiana?
New Orleans?
New Orleans. Thank you. It's been a long day. That, hopefully there wasn't a gravitational wave passing through at the same time, because what are the odds that we would be able to detect it if there's all this probable noise
that they normally don't cancel out.
Right.
You would not be able to tell, right?
Right.
Their detectors would be showing you all kinds of fringe patterns.
Yeah.
Right?
Now, I don't know for sure. Maybe they do have something in place.
And they're like, we can withstand hurricanes.
Yeah.
I mean...
Possibly. I mean...
I wouldn't...
I wouldn't put it past them, because it seems like they put a lot of thought into everything here. But I suspect that there was probably some interference from that, storm that would cause them not to be able to detect it. But yeah, just an incredible amount of things that go into making this work, and making it operational for the
going
purpose.
Going from the idea to the actual realization of the apparatus you actually need. Pretty cool.
So I'll continue on here just to kind of showcase where the other two gentlemen play their parts here. Quote, a few years after the developments at MIT, another interferometer prototype saw first light at the California Institute of Technology, or Caltech, under the auspices of theorist Kip Thorne. During the 1970s, Thorne and his research group worked with great enthusiasm on the theory of gravitational waves and their astrophysical sources. Their predictions of the expected signals from various astrophysical events played a decisive role for the design and funding of LIGO, and for the first steps to prepare the online and offline analytical tools enabling breakthrough discovery of GW 150914. In the late 1970s, Thorne succeeded in convincing the Caltech leadership to create an experimental gravitational wave group led by Drever, recruited from Glasgow in 1979, and Caltech's Stanley Whitcomb, end quote.
So Kip Thorne has been studying this since the 1970s.
Long time. A lot of thought goes into something like this, right?
Yes, it does.
Yeah. And then finally, quote, to ensure the successful transition from a prototype activity to a realistic big science venture, Caltech, in consultation with NSF, appointed Barry Barish, who had leadership experience from large particle physics projects, as LIGO director starting in 1994. Barish transformed LIGO from a limited MIT Caltech endeavor to a major international gravitational wave project. Barish's aim was to construct LIGO in steps, with the first stage, initial LIGO or iLIGO, providing a verification of the technology, and, in the best case, offering a possibility to detect gravitational waves. The second stage, advanced LIGO or aLIGO, would be based on more advanced technology and reach a sensitivity that would make detection of gravitational waves probable. The construction of the facilities at Hanford and Livingston proceeded under Barish's leadership from 1994 to 1998. He then oversaw the installation and commissioning of LIGO's initial interferometers from 1999 to 2002, and the first data-taking runs 2002 to 2005, end quote. So Barry Barish seems like just the guy to be the man, right? Like, he was the one that was like, nah, we're doing this. This is happening. This isn't going to be some idea that eventually gets defunded and the whole thing gets converted from underneath me. He was like, nah.
Yeah, he convinced enough people.
We're doing this. This is happening.
Yeah, he convinced enough people that we need to do this.
Yeah. So, bravo. I think we're going to skip the last piece here that I have written for gravitational waves. Just in the interest of time, I want to kind of make mention for the Nobel Prize in Chemistry, and we're approaching the end here, Tom. Actually, this is
I had a feeling because I wanted to talk a lot bit about them, but so we can call that on gravity waves.
Yeah,
I think we discussed them quite a bit, and then just paint a picture of what was going on for the Nobel Prize in Chemistry.
Yeah. So this one, I'll be honest, I know the least about, just in terms of like
the actual science that goes into it.
Yeah. So the Nobel Prize in Chemistry 2017 was awarded to Jacques Dubochet, Joachim Frank, and Richard Henderson, quote, for developing cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution, end quote. We have the illustration to bring up. So these are our gents here.
And I just want to quickly point out that the most amount of people that can be awarded the Nobel Prize for one award is 3 people.
Correct.
Apparently.
And they must be living.
Yep, they have to be alive.
Yep. And there's some other rules with it.
There was also some more slight controversy out of no more female winners again, and the amount.
I saw
a lot
that
of articles about that too.
Yeah, I saw some of those articles. We'll leave that for another time. Maybe that can be a Tom's Corner one week or something. Cryo-Electron Microscopy, Tom, is a technique for looking at the smallest of small-scale structures. While the electron microscope has been around in some capacity since the 1930s and can be used for analyzing inanimate objects with a high degree of resolution, only recently have key developments allowed the technique to become applicable to studying biological material. In order to understand the challenges associated with using an electron microscope on biological material, you first need to know how an electron microscope works in general. We can bring up an image and very quickly kind of decipher that for you guys. So your electron microscope, you basically have at the top some sort of electron gun, they call it, but you can think of it kind of like a CRT, like your old cathode ray tube televisions that would fire electrons to produce the image on the screen. That beam of electrons is then going to get focused down into a lens. The lens then directs the electrons in a straight line so that they interact with the specimen, and then they'll bounce off the specimen, they'll scatter, and hopefully get detected by a detector that sits above the beam there.
And it's basically the strongest type of microscope we have, right?
Correct.
So that's why it's so good for viewing small things, but what made it difficult to view biological material?
So biological material is susceptible to being damaged if you're
by the electron microscope itself, by
right.
trying to view it.
If your electron beam is firing high energy electrons, you can substantially change the sample that you're trying to look at. And so you need to use lower energy electrons to make sure you don't do that. But then the problem with that is that biological material does not typically have a high degree of contrast, and so it makes it
hard to actually see it.
Right. It makes it hard to distinguish what you're looking at when you finally do create an image of it. Not to mention that your sample also has to be encapsulated in a vacuum chamber. And when you do that with biological material, a lot of times the water that is involved in your sample will, basically
like leach out. Almost.
Yeah, you lose the water
because you have no atmospheric pressure or anything, and it's a living thing. It sort of fudges it all up. That's the best way to say
exactly.
it. Right?
And if you lose the water, then again you're changing the
structure of what you're actually trying to look at.
Right.
Okay.
So there were some thoughts of how we could solve these problems, and cooling the specimen samples was one of those solutions that they proposed, because cooling the water will then reduce the chance of evaporation in the vacuum, and it should help the biological material be protected from electron radiation. But freezing the water will cause it to change into ice crystal
structure, yeah.
Right, which again changes the structure of the sample, and so that there were more complications, right? X-ray crystallography, a technique that is still widely in practice today, was theorized to solve some of these problems, but you can't really use that so well with biological material, because you need to get, let's say you're studying proteins, you need to get proteins that are all entirely symmetrical, and then you have to shove them into a crystal structure in order to scatter the light that you're looking to then analyze and reconstruct the 3D image of what you're looking at. And so that doesn't really work so well for large proteins. It doesn't really work so well for, if you're interested in how a protein moves and operates over time, then having them all oriented the exact same way doesn't,
like frozen in place, yeah,
doesn't help you learn anything from it. So Richard Henderson was kind of the first guy involved here, and he made progress by figuring to use some sort of a glucose solution instead of water, right?
Okay.
This worked a little bit better. To quote here the document, Henderson and Unwin replaced water with glucose solution to preserve samples in vacuum and used the new preparation method in studies of intact protein crystals. right? So he was able to kind of reduce the amount of damage that things were taking. He also did this with a particular protein called bacteria rhodopsin, and was able to kind of tilt the electron microscope or the sample within the electron microscope in order to get a whole bunch of different 2D platforms, and then by way of a technique called Fourier transform, he was able to remodel them into three-dimensional shapes again.
Okay.
So that was a big thing here. Joachim Frank tackled the problem of low resolution, right? Because even with Henderson's contributions, there was still this low resolution problem. Quote, Frank and colleagues presented a method for aligning low-dose images of individual molecules using cross-correlation functions. A quantitative analysis of the problem was presented in 1977. The analysis concluded that it would be possible to locate randomly positioned particles using non-destructive electron doses. Consequently, the implication was that it would be possible to average images of many radiation-sensitive particles to eventually obtain high-resolution data. The feasibility of the approach was illustrated in studies of negatively stained glutamine synthetase, end quote. So he kind of went into the mathematical theory of, all right, how do we average over a bunch of different samples to get a high-resolution image of one, right?
Gotcha.
So that was pretty profound. And then finally, Jacques Dubochet addressed the cooling problem, where, quote, in 1981, Dubochet and Alasdair McDowell finally presented a method that allowed formation of a film of non-crystalline solid water on a specimen grid for observation in the electron microscope. Water was sprayed on a carbon film mounted on a grid, after which the grid was rapidly immersed in liquid ethane or propane at about negative 190 degrees Celsius, maintained by cooling in liquid nitrogen. The thin layer of vitrified water was shown to yield nearly uniform absorption of electrons in the cryo-electron microscope, end quote. And so all that said, Tom, right?
Yeah, so they, I think a nice way to describe it is that they really needed to figure out step by step what they need to do mathematically and physically to the biological material to allow an electron microscope, which is our most powerful small viewing tool, to view biological material so we can actually see those structures and see how they work in the body.
Correct. And now we've gotten to a point where the electron microscope, cryo-electron microscopy, is a technique that works just as well as X-ray diffraction, in a sense, right? And so we're going to bring up an image here. Credit goes to nobelprize.org and Martin Högbom for basically an illustrative way to look at what our resolution of biological materials, such as protein structures, looked like prior to these discoveries being implemented back in, before 2013, and now what the resolution looks like at present, right? So this kind of one protein structure goes from having low resolution to high resolution if you look from left to right. And this is really important because we have all these synchrotrons set up, right? Brookhaven National Lab has the NSLS-2, right? The National Synchrotron Light Source 2, which is used in some capacity to do X-ray diffraction on protein structures in order to get a better idea of what their shapes are. But this technique, Tom, is basically going to render that unnecessary, in a sense.
Yeah, that's pretty wild.
That's pretty crazy. What does that mean for the future of user laboratories like Brookhaven National Lab? I don't know yet.
Yeah, because the NSLS-2 is an amazing piece of technology.
Absolutely. And I think that there's still obviously going to be some utility for it. It's not like the only thing that they sample there are protein structures and biological material.
Of stuff, yeah.
Ton of different science that you can do with that. But I find it interesting that probably a lot of people studying biological material in this way will probably opt not to use a synchrotron because they can just learn more from using cryo-electron microscopy. You know, if you wanna see things in action, you can do that with this technique. You can't necessarily do that with synchrotron radiation.
Yeah. And I think moving from there, once you do get that higher resolution and sort of can see how the exact structures interact with other proteins and molecules, I think that I read an article that that's really the hope for the future of developing pharmaceuticals and other things, is that right now we kind of just know how systems work and we sort of try and influence the systems with pharmaceuticals, but they're hoping that in the future that they're targeting extremely specific things like the shape of receptors in proteins and other parts of your body that they're hoping to learn from studying these three-dimensional structures.
Right.
So they're hoping that that sort of follows over to the pharmaceutical side of the industry.
Yeah. I think it'll be possible as long as technology can keep up, right? The big problem is that we don't have processing power in computers that are able to resolve all the possibilities of the structures in any given protein or DNA, product.
So yeah, it's still ongoing research that involves multiple fields that's looking to keep trucking forward.
But yeah, I think it'll come. All right, Tom.
So those are the three.
Those are the three that matter for this show, at least. Obviously, congratulations are in order to all the recipients. Congratulations are in order to all the recipients that we didn't discuss, and you guys can obviously go check out everything else that was awarded in terms of the other topics there. Tom, that's going to do it for this week on trèSci Weekly. A little bit longer of a show, but
just a few minutes.
That's okay.
Wanted to give time to those awesome experiments and science.
Right on. So, Tom, I will see you next week for episode 36.
Sounds good.
And I'll see all of you guys next week for episode 36 as well. And Mush will be there. That's it, guys. See ya.